By definition, genetics is the study of genes. But let's start a little smaller than genes and work our way up. There are 4 major macromolecules of life: carbohydrates, lipids, proteins, and nucleic acids. Nucleic acids are DNA and RNA, where DNA stands for deoxyribonucleic acid, and RNA is just short for ribonucleic acid. DNA and RNA are like the words that make up a recipe of how to make every living organism that ever was or ever will live on our planet. The information contained in DNA and RNA molecules determines all the traits we see in living organisms. Now, we can talk about traits in both a genotypic and a phenotypic way. Phenotypic traits are expressions of the genotypic information. For example, a phenotypic expression of a particular sequence of DNA in a human would be brown hair. The information contained within my DNA, called my genotype, influences what color hair I will naturally have. It's important to note that not all phenotypic expressions of genotypes can be seen with the naked eye. It's easier to talk about those we can easily see, like hair color or foot size, but phenotypic expressions can also be things like cystic fibrosis, which is a genetic disorder. DNA and RNA are complex molecules made up of nucleotide base pairs that come in the flavors adenine, thymine, cytosine, and guanine for DNA. For RNA, we see similar nucleotides, but instead of thymine, there is another compound called uracil. This takes the place, leaving RNA with the nucleotides adenine, uracil, cytosine, and guanine. In eukaryotic organisms, the DNA is found in the membrane-bound organelle called the nucleus inside the cell. DNA is the most abundant form of genetic information found in complex organisms. For prokaryotes, prokaryotes can have either DNA or RNA, and this is going to be found in the nucleotoid region, since by definition, prokaryotes lack a true nucleus. DNA is a double strand consisting of 2 complementary strands, and this is the shape where we're going to refer to it as the double helix. So think of a ladder and visualize it just as we're about to climb. It's like an old, wooden, creaky ladder that you have to lean against something before you can climb it. It has 2 long vertical sides and many shorter horizontal bars in the middle. The 2 long vertical sides are what we're going to, in our molecule, call sugar phosphate, and this is going to be the backbone that gives strength to the molecule and acts as a scaffolding for the nucleotide base pairs to latch onto. The rungs of the ladder, where you would put your hands and feet, these are going to be those nucleotide base pairs. That double helix shape only comes when we take that ladder and we twist it. This shape allows for an enormous amount of information to be stored in a very tiny amount of space. And just like music notes on a sheet are very specific in terms of their pattern in order to create a song, or how letters on a page that make up a poem are going to be very specific, adenine, thymine, cytosine, and guanine are the notes and the letters of how to create life. We call these compounds base pairs because they will pair in a very specific way. Adenine will pair with thymine. Cytosine will pair with guanine. A with T, C with G. And their sequence is going to be extremely important, but only in the sections of the genome that are actually coding for something in the current version of a particular species. These active sections are called genes. Think of genes as a sequence of notes that makes that beautiful song. They are the letters in that particular form in just the right way that allows us to understand and ignite our imaginations in a powerful poem. If they were in any other sequence, they wouldn't make sense. They wouldn't spell words or make pleasurable sounds, thus rendering the piece completely useless. There's a lot we don't know about the delicate ways that genes work, behave, interact with the world. But as dedicated researchers have even more access to advanced technology, who knows what we're going to find in the vastness of living cells. Until next time, thank you for joining me on Exploring Environmental Biology.